get started. We have a jam-packed day, so I want to make sure that we remain somewhat on time. There we go. So welcome to our 8th annual Heart Day. It's hard to believe we've been doing this for 8 years now. Something that Dr. Ramaswamy and I started 8 years ago in BME. And this is the signature event of our Heart Month lecture series, which goes throughout February. We have cardiovascular, neurovascular, perivascular researchers come and give talks. And the original goal of this event was to kind of bring South Florida cardiovascular researchers together to discuss what's going on locally, and then also, of course, bringing a keynote speaker to discuss. And it was a very small first event over in an engineering center. It's really blossomed where today we're expecting over 100 people to come throughout the day. We have a high school outreach session with some 70 some odd high school students who will be here participating in some outreach. We had 45 abstract submissions this year for posters and presentations, so quite outstanding. We also have really expanded, and we have people here today from Baptist, showcasing the strengths of our collaboration with Baptist and Miami Cardiac Vascular Institute. From the University of Miami, we've got people coming down from, of course, St. Lucie, from the Center for Translational Sciences. So really an exciting event and a testament to the existing strengths in cardiovascular research and medicine, which I think is going to serve as a nice foundation for our future growth as well. Before we begin today, I want to thank Florida Heart Research Foundation for their continued support of our research and our educational programs, which will be spotlighted all throughout the event today. And I also want to thank the Department of Biomedical Engineering and our Wallace H. Coulter Foundation Endowment, which has paid for this event. Special thanks to their outsides that can't hear me, Vanessa, Ingrid, and Claudia, who put together the amazing abstract books and everything that you see today. Have done a lot of work doing that. Also special thanks to Lucas and Anthony and the rest of Healthy Hearts Club, and Bailey and Mays from the FIU CDSEC for organizing the outreach event. It's going to be fantastic. So I want to begin by welcoming Dr. Andres Hill, who's the Senior VP for Research and Economic Development and the Dean of the University Graduate School, to provide us with some opening remarks.
Thank you, Josh. I want to thank Josh for his leadership in building cardiovascular research at FIU. I think it's a great partner, and the things that we are doing and the future growth of cardiovascular research here at FIU. I want to welcome the keynote speaker and all of those that may be here from outside of FIU and maybe here later. We will be passing around since this event is in the building where my office is, so it will be easier than that. I want to have a special welcome to all the students. I understand from Josh that we have here both undergraduate and graduate students. They're all important to me, but the graduate students are especially important as dean of the graduate school. I'm going to say a few words that hopefully are not going to get the students nervous if you have not heard the buzz of everything that's going on with the funding of research in the United States in terms of things that are going on with the NIH and other agencies. If you have not heard anything, that's fantastic. Focus on your research and focus on, so don't get nervous for that. But just in case you have, obviously there is quite a possible shift on how the federal government funds biomedical research and all types of research. I'd say that don't panic when you hear certain news, because my message to the students and the faculty is that we keep going in the direction that we're going. Sometimes there's a piece of news that certain research is not going to be funded anymore, and then the next day they tell us that it's getting funded. So we're only reacting to the specific news that we have on specific grants. In terms of students, I understand that there are some of the graduate students, but all of our PhD students for the most part are supported either from grants or from the college and the graduate school. If there's any grant where a student has an assistantship from the grant, and that grant is either the funding is reduced or part of it is taken away, that we will do that, okay? I don't think that the area of research of the group that's sitting here is at any particular risk of, grants ending. We've had grants from USAID that have ended, right? But there are other things going on in terms of how grants are funded, that there may be some reductions on that. But we shall continue in the direction that we're going. We are a university that has been growing our research, and we have a new strategic plan that will double our research in 5 years, and we're continuing that. So just in case, if this is the first time that you hear about this, then forget what I told you, right? Don't come out here getting nervous. So I want to just say a few words about this event and cardiovascular research at FIU. Obviously, as Josh mentioned, this is the 8th year, so it's kind of funny seeing I go to events when it's 10 years, 15 years, and then I realize I'm getting older, right? Losing my hair, etc. So we are also delighted that there will be partners from Baptist Health here, because we have entered into a partnership with Baptist Health that is going to impact clinical services in South Florida, but also research. And yesterday, we entered into an agreement with Nicholas Children's Hospital. So basically, Baptist and Nicholas will be our main hospitals. We still have relationships with other healthcare systems throughout South Florida. Florida Heart Foundation is providing a generous gift to, and it's not new for them. They've been supporting cardiovascular research here at FIU, and for the next 10 years, it's about over $1 million a year, basically, that they will be supporting cardiovascular research. We're going to create a research center around that gift, and that center will report centrally to the Vice President for Research, because we see the cardiovascular research as an interdisciplinary enterprise that will involve not only biomedical engineering, but will also involve other colleges. As Josh mentioned, there will be people here from CTS, which is a facility that FIU has about 2 hours from this campus, that used to be the Torrey Pines Biomolecular Institute. And it's the Center for Translational Science for us, and there we are recruiting researchers, scientists that do work around pulmonary and cardiovascular and cancer. So that will be another component of the growth that we have on cardiovascular research. All of the students probably spend most of the time at the engineering center right now, I'm assuming, but I'm assuming that you can see the new building, one new building that is at the corner of 107th and 8th, the Engineering and Innovation Complex. There's going to be a second building there, and we will have this cardiovascular research center in that new facility. I think that facility will probably sometime, that first one is completed, so we're going to start the process of setting up the center there, and that will be brand new and state of the art, and it's part of the growth that we want to have around biomedical engineering and relationships with the medical school and with Baptist Health. So I think that our future is bright in this important area of research. I've had a health situation 3 weeks ago where I ended up, I was in Tallahassee in an ambulance. I think everyone was thinking that I was having a heart attack. Okay? It was that, but you realize, the importance of dealing with heart disease in terms of the health profile in the U.S. So I see that there will be a lot of opportunities for us to grow in this area of research, and these kind of gatherings are important for collaboration, which is the key to success in all research, and particularly in this area. So again, I hope that you have a great day today, and I'll be stepping by now and then during the morning. And I want to again thank you, Dr. Hutchison, for all the work beyond all the things that the faculty member has to do in terms of their own lab, their research, and their teaching. He's really been a leader for us, and will continue to be a leader for us in growing cardiovascular research here at FIU. Thank you.
Many thanks to Dr. Hill. He's really championed our efforts to build cardiovascular research at FIU. The headwinds were sometimes pretty strong in that regard, and he was a champion all the way through. So that's very, very much appreciated. So now I'd like to introduce our keynote speaker for the morning, which is Dr. Jessica Wagenseil, who's a professor of mechanical engineering and vice dean for faculty advancement at Washington University in St. Louis. She's an internationally recognized leader in cardiovascular mechanics, with particular focus on development and microstructural patterning of cardiovascular tissues that dictate their function. She received the Yozo Award from the American Society for Matrix Biology in 2021, recognizing her efforts in elastin and dictating arterial mechanics. And she also received the Emerson Excellence in Teaching Award in 2023. She has been elected as a fellow of both BMES and AIMBE, both of which recognize leaders in biomedical engineering in particular. Her work has been well funded by the National Institutes of Health, the National Science Foundation, the American Heart Association, and the Marfan Foundation, and we are incredibly honored to have her here today.
All right, thank you so much for that nice introduction, and I am happy to be here. Happy Valentine's Day to all. I'm very happy to be spending mine in a place that's 60 degrees warmer than my home, so thank you for inviting me here. And it's just a real pleasure to see, all of this cardiovascular research happening here. I'm excited to listen to the flash talks and see the posters, and really hope to, start some collaborations. So as Josh mentioned, I'm from Washington University in St. Louis, and I'm going to talk to you today about biomechanics of thoracic aortic aneurysms. I'll start a little bit with my background. So I am from Nevada City, California. It is a tiny gold mining town that is downtown. It's an old gold mining town, and it's a cute little tourist trap for people from the Bay Area to go to, and not a lot happening, but absolutely gorgeous surroundings. So when I went to undergrad, I moved to University of California, San Diego, and my major was in bioengineering. And I'm old enough at the time that people were like, bioengineering, what's that? And you're not going to get a job. Of course, you all know, right, that BME has taken off, and we're all going to find great jobs. I then, for my PhD, went to Washington University in St. Louis for a PhD in biomedical engineering. I then stayed there for a postdoc in cell biology. So I had this training in kind of biomechanics, and then also in cell biology, and specifically matrix biology. My first faculty position was at St. Louis University in biomedical engineering, and then I came back to Washington University in 2013 in mechanical engineering. The work that I'm going to tell you about today was done in my lab, and this is a picture from last summer. And my lab is a combination of postdocs, of graduate students, and undergrads. And we typically have a bunch of summer undergraduate researchers. And I'm thrilled that one of them, I don't know where she is, or she's in the back, Mariana Vizo, who's visiting here from UM today, was one of my summer undergrad researchers 3 summers ago or so. So super excited to then see where people go and what they do after they spend some time with me. So we study vascular mechanics, and we particularly study the aorta. This is an ultrasound of the mouse aorta, and a pulsatile cardiovascular system. So we all have pulsatile cardiovascular systems. Invertebrate animals do not, but vertebrate animals do. And the protein that I am most interested in studying, which is elastin, actually comes about in the vertebrate cardiovascular system and came about in evolution when we had pulsatile blood pressure and flow. So elastin is actually these red elastic fibers here. So in a vertebrate aorta, it's organized in these layers that we call elastic lamellae. And you can see that they alternate with cells. The little turquoise are cell nuclei. And then we also have collagen, which is shown in green here. Collagen's mostly out in the adventitia, and there's a little bit here in the media. And then elastic fibers are mostly in the media. So elastin and elastic fibers provide this elasticity to the aorta that allows it to stretch and then recoil every time your heart beats, which is about once a second in humans sitting here. And about 10 times a second in a mouse, which is what we study. The elastin and the collagen together provide the passive mechanical behavior of the aorta. And it's typically viewed that elastin is responsible for the resistance to stretch in the low stretch region, and that collagen is responsible for the resistance to stretch in the high stretch region. So the collagen is typically buckled or tortuous, and so it doesn't contribute until you get it straight. And then it contributes and kind of locks the aortic wall so that you don't over-distend and damage the cells within the aortic wall. And then elastin's in this region and provides that elasticity. In the physiologic range, which is where these blue and yellow parts overlap, that's where a normal aorta operates. So we're operating in a range where both elastin and collagen are contributing to the mechanical behavior. But in many of the diseases that we study, there are defects in the elastic fibers. And so how these two things contribute shifts. So you can change the mechanical behavior of the aorta, you can change how elastin and collagen are contributing to the load. And the disease that I'm going to talk about today are thoracic aortic aneurysms. So this is an image of the ascending aorta. So this is the heart. This is the ascending aorta in a human patient. And your ascending aorta is supposed to be about 2.5 centimeters, or about 1 inch in diameter. So this is 4 centimeters, much bigger than it's supposed to be. And if you have an aorta that's about 4 centimeters, you will most likely be getting surgery. So they will replace it with a graft because they're worried that it's going to dissect, which would be a little tear in the wall that blood can get into, or they're worried that it's going to rupture. You do not want your aorta to rupture if you're not in a hospital under care, right? Because that is taking all of the blood flow from your heart. So in this case, this patient is going to get surgery. This is the thoracic aortic aneurysm in the ascending. And then you can see in this picture, so here's the heart again, here's the ascending. This patient also has some dilation in the descending aorta, but also always in the thoracic, which is the chest. And then you can see some tortuosity, and then this would be the abdominal aorta. So you may have also heard of abdominal aortic aneurysms, but everything today I'm going to talk about is thoracic aortic aneurysms. And about 20% of those are caused by genetic mutations. So we know that the patient has a mutation that leads to this disease in about 20% of the cases. And many of those affect elastic fibers, which is what my lab is interested in. And in human patients, one of the characteristics, regardless of which mutation one might have, or even if we know that it's caused by a mutation, but elastic fiber fragmentation or degradation is characteristic in thoracic aortic aneurysms. So these black lines are the elastic fibers in a human aorta, and then you can see these areas where they're highly fragmented or missing, and actually replaced by other matrix materials. And so this kind of defect is associated with this disease, and it affects the mechanics due to what I just told you about how elastic fibers and collagen contribute to the mechanics. So I have to introduce a little bit about elastic fiber protein assembly because we use mouse models that have genetic mutations in some of these different proteins. So they have genetic mutations in the genes, and the proteins are involved in the assembly. So this is just a little cartoon of how elastic fibers are made. The soluble tropoelastin is made from the cell. It's cross-linked by lysyl oxidase. It interacts with some helper proteins, 54 and 5. It then collects on the cell surface and is transferred out to these microfibrils, which have have mostly fibrillin 1, and then you end up with this whole structure that makes those layers of elastic laminate in the wall. And so today I'm going to talk to you about mice that have genetic defects in lysyl oxidase, in fibrillin 4, and then also in fibrillin 1. The other thing that's important about elastic fiber assembly is there's a saying that you're only as old as your elastic fibers. So the half-life of elastic fibers are about 70 years. So they need to last more or less most of our lifespan. And so understanding how degradation affects the mechanics is important because we're typically not making more of them. So many of you might study proteins that turn over on an hourly, daily basis. This is a very different case. These are not turning over on an hourly, daily basis. So degradation of them is a real problem. So the overall theme of what I'm going to talk about today is that elastic fiber defects alter the wall stresses, and then also transmural transport, and that they contribute to aneurysm progression. So this is one of our mouse models. This is down here. It has elastic fiber defects due to a mutation in fibrillin 4. This is a normal aortic wall and a mouse aorta, so lovely black layers of elastic fibers. And then here you can see that those black layers are less intense and they're fragmented. So when we have these changes in mechanical properties, we change the way that the stresses and the stretches are experienced by the wall. And the stresses in the wall come from a variety of sources, including the circumferential stress, which I'll talk about a lot today, in terms of the pressure that's on the wall, and that's related to the radius and the thickness. There's also axial stresses, and so the blood vessel is tethered in the system, so there's axial stresses along the length. And then there's also shear stresses due to blood flow, which I'll talk about too. And all of these equations in this diagram are highly simplified, right? So the actual way of calculating these stresses is much more complicated and requires computational models, and we do some of that as well. The other thing I'll talk about today is transmural transport. So this is your aorta. Inside the lumen is your blood, which has a concentration of cytokines. It also has a pressure, right? Humans and mice have an average blood pressure of about 100 millimeters of mercury. And so this gradient of both concentration and pressure can drive things across the wall. And so we're interested in how things might be driven across the wall and how that might be altered if, in this case, my little green elastic lamellae are fragmented or damaged. And this is not just a one-way street, right? When you have these changes in the mechanics or changes in this transport, we think that these feedback then that might cause even more elastic fiber fragmentation or degradation in a positive feedback cycle that then continues to advance the disease. I want to start by talking about the mechanics and the wall stresses. So when we're looking at solid mechanics of the vascular wall, we use this commercial system here to test mouse aorta. We mount, in this little bath, we mount the mouse aorta, which you can see here. And in this diagram, you can see that we can stretch it along the length, and we can measure the forces, and then we can also inflate it and measure the diameters. And from this information, we can get the biaxial mechanical properties of the mouse aortic wall. We also need unloaded dimensions because you can't calculate stresses and strains without knowing the unloaded condition. And so we cut rings and we get the unloaded dimensions as well. I'm going to start by telling you about some work in our mouse with fibrillin 1 underexpression. These are a model of Marfan syndrome, which is associated with thoracic aortic aneurysms, and then also with some bone phenotypes and some metabolic phenotypes. This is a CT scan of the entire aorta in a wild-type mouse. This is the ascending up here, and then this is the ascending in what we call the MGR mouse model, which is this reduced fibrillin 1. So you can see that they have aneurysms. These mice also have spinal defects due to some of the bone phenotypes of Marfan syndrome, and so they have kyphosis or hunched back, and so you can see that in the aorta as it follows the spine. They also have fragmentation of the elastic fibers, which you can see down here. So layers of black in the wild type, and then much less intense staining of the layers in the MGR, and then also the fragmentation. So these mice that you'll see in my slides are just labeled mutants. They develop thoracic aortic aneurysm sometime between 1 and 4 months of age. They die of cardiovascular complications by about 6 months of age. And the cardiovascular complications is typically related to aneurysms, but it's there's no difference in the incidence of Marfan syndrome or TAA. So males and females get this at about the same rate, but males actually have a worse outcome, and we see that in the mice. So we were interested in trying to understand the mechanics of these, and also perhaps, what might be going on in differences between males and females. So the first thing we did was track TAA formation over time. And so we used the ultrasound machine that we used to take that first video that I showed you. And this is the ascending aorta in the wild type. This is the ascending aorta in the mutant. And you can see that this is just measuring the diameter at this location, and it's bigger in the mutant than the wild type as the aorta dilates. So if you look at this, we are tracking the systolic inner diameter up here, the diastolic inner diameter down here, and in all of the data, the males will be on the left, the females will be on the right, and the wild type is always in blue and the mutant's in pink. So what you can see is we see this dilation over time in both males and females. But if you look at kind of the trend of how this works, the males have this continual increase and steep slope in dilation over time, whereas the females dilate and then kind of reach a steady state. We take this data, as we have those systolic and diastolic diameters, and we know the pressures of the mice, and we calculate stiffness. So stiffness of the ascending aorta has been directly linked to adverse cardiovascular outcomes. And so we're interested in how stiffness might play a role in this disease model. There's two different stiffnesses that we can track. One is called structural stiffness, which depends on the material properties and geometry, and is directly related to how much that aorta expands and recoils in that video that I showed you in the beginning. There's also material stiffness, which is directly related to the material itself, but not to the geometry. So you could imagine if you have a tube, right? If the tube is made of the same rubber but has different geometries, like thicker, it's going to have different structural stiffness, but the same material stiffness. So when we look at that over time in our mouse model, so starting with the structural stiffness up here, you can see again, it's much increased in the mutant mice, and it increases in both the males and the females. And then this material stiffness, again, increased in the mutants compared to the wild types. But in the material stiffness, at least, we have the same trend, where males continue going up, and females have a much less slope over time. So they're not increasing as much as they get older. We then do our mechanical tests. So we take the aorta out. We do a variety of mechanical tests on it. Here I'm just showing you one protocol of our mechanical tests. So we set the aorta at its in vivo length, and then we inflate it and measure the pressure at the bottom and the diameter at the top. At 1 month, so the pink curve for the mutant at 1 month is down here in all the blue curves. So at 1 month, you can't really see the dilation, not much differences in the pressure diameter behavior. But then you can see as we get up to 2, 3, 4 months, we have increases in the diameter. You can also see how big these error bars are, right? So very different range of how much dilation you get as it grows. We calculate our stiffness values again using this ex vivo data. And consistent with our in vivo data, we have a much higher stiffness in the mutant mice than in the wild types. And this trend in the material stiffness here, where the male is increasing quite a bit, and the female is increasing at a much slower rate. We take this data and we do stretch and stress calculations so that we can get that curve I showed you in the very beginning, where we have a stretch and stress curve that shows the mechanical behavior and the material behavior of the aorta. We make some assumptions so that we can use these pretty simple equations. And then we can get these representative curves that look similar to what I showed you in the beginning, where it's nonlinear with low stretch regions, and then it becomes stiffer at the high stretch regions. And we do a pretty simple analysis on these curves to try to understand how the mechanical behavior may be related to that elastic fiber fragmentation and any remodeling due to collagen. So we just fit what we call a low modulus and a high modulus. So to the lower region of the curve and then the higher region of the curve to understand how these slopes are changing. And when we do that, the low modulus is reduced in the mutant animal. So the part that we might attribute to elastin in this low stretch region is lower. And then the high modulus is increased. So the part that we might attribute to collagen is higher. And I really like this analysis, even though it's simple, is because a lot of people I talk to are like, is the vessel stiffer or less stiff? I'm like, well, vivo, if we look at the in vivo loading, it is actually stiffer. So because those were all those structural material stiffnesses that I showed you. And again, kind of harping on this trend that we see large changes in the males, less changes in the females, and especially when you get this high modulus that we think might be attributed to collagen. So we went looking at the wall structure, and these are multiphoton microscopy images of the wall. The cells are stained with DAPI and glue, but multiphoton is really nice because you don't have to stain elastin or collagen. So structural properties of those proteins provide a signal, and you can just get images without any antibodies. So elastin is in green, and then the collagen's in red. And so a male wild type's up here at the top. You can see not much changes in the wall structure over time. And then the male mutant, so if you go down the 1 month column, you can't really see a ton of differences. But as it dilates over time, especially the male mutant here, you can see by 4 months we have much less intense staining of the elastic fibers and a high degree of degradation or fragmentation. And then if you look down here at the females, you do still see some fragmentation, but it's certainly not as bad as it is in the males. If you quantify this data, and we calculated endpoints, so the kind of times you see a fiber just end in our field of view, lots and lots of endpoints in the males, a little bit more in the females, and then intensity, same idea. So intensity is dropping down significantly in the males, and a little bit lower over time in the females. So the same trends that we had seen in our mechanical parameters are showing up in these microstructural parameters. So collagen, this is a cross-section of the aortic wall, and collagen's a little hard to see because there is collagen, which you can see in the fragmentation in the media, but it's hard to see when there's so much elastin in the media. And then in the adventitia, it's a little bit variable depending on how much you capture. And so to get a better look at the collagen, we do on-FOSS imaging. So we take a piece of the aorta and we actually step through its thickness so that we get kind of a planar view of what the collagen looks like. And it's a little hard to tell from the individual images, but if you quantify this, and we're just looking at intensity in what we show as the medial region, what we show as the adventitial region, you can see that the collagen intensity in the males is increasing, and then the adventitial collagen in the males is also increasing. So again, consistent with this idea that we have fragmented elastic fibers and that we seem to have collagen being deposited to make up for some of that. So from the study, we have kind of lots of what I would call descriptive data on this mouse model, but interesting changes in time and with sex. And so what we kind of concluded is we see this ever increasing values of stiffening and the ECM remodeling in the males, and then the dilation just keeps increasing and increasing. Whereas in the females, something is causing more of an asymptotic relationship, so it doesn't keep going. And then we know from both our studies in this study, as well as other people's publications, that males have a much shorter lifespan. So the mean lifespan for the males in the study is like 3 months, and then much longer in the females. The mean lifespan of the females is about 6 months. And then we're also, if you think about it, because of that lifespan, we're actually biased in our study towards males who lived, right? So we have 4-month data, and we only have that data on males who lasted that long. And so we think that some of this interaction between the stiffening and the changes in ECM may be related to outcome. Of course, we have no idea why, but there's a couple hypotheses out there in the literature. So sex hormones, in particular estrogen, have been shown to be protective, and so that may play a role. And there's also sex chromosomes. So one of the, it's called a tissue inhibitor of matrix metalloproteinases, so basically controls how ECM proteins are degraded, is actually linked to the X chromosome. And so female mice might have more of that. So I have a postdoc now who's looking at some of these mechanisms and why that may play a role. But of course, we also want to eventually be able to either predict progression or outcomes, so that if you have an aorta that's 4 centimeters, we know, should we do surgery today, right? Or can we wait a little bit longer? And so the couple things that our study has shown is that stiffness may be related to outcomes. And there are ways that you can measure stiffness in humans. So people look at something called pulse wave velocity, which is related to the aortic modulus, and you can measure that pretty easily in humans. And the other thing is if you could measure this elastic fiber degradation or some of that collagen deposition. So we're certainly not going to be able to take out a little sample in a living human patient very well, but there are like some MRI imaging techniques that can tell you about organization of the matrix in the wall that may also be useful in terms of trying to predict how things are going to go. The other thing we'd love to be able to do is prevent this elastic fiber degradation and the TAA formation to begin with. I told you these elastic fibers need to last you your whole lifespan, so if we can stop the degradation, maybe we wouldn't have to deal with the problem of making new ones. So one of the areas that we've been working on is looking at this small molecule called PGG for elastic fiber stabilization. So PGG is similar protective ability to tannic acid. It's actually like this whole structure. This whole structure is tannic acid, but then this middle region is PGG. Tannic acid comes from tanning hides. So it's how you make the leather in your shoes if you happen to be wearing leather shoes. Right? So you take a hide, you treat it with tannic acid, it cross-links elastin and collagen, stabilizes it, and you have leather. You don't want to treat cells with tannic acid, not going to be happy. But PGG is actually non-toxic. And so about 20 years ago, it had been shown that if you treat rats in an abdominal aortic aneurysm model, so at this point, at time point 0, they treat the rats with PGG or nothing, just saline, and then they treat it with something that causes an aneurysm. And in the saline treated, right, you get a large increase in the diameter, and you get fragmentation here of the elastic fibers. And in the PGG treated ones, you get less increase, and you get less fragmentation of the elastic fibers. And so this was in a rat model of the abdominal. We wanted to know, how does it work in the aorta? How does it work in a mouse? And how is this really changing mechanics? Because there weren't mechanics studies in this publication. So this is the project that Mariana actually worked on when she was in my lab. And so we took the mouse aorta. We had an untreated case, and we did a bunch of mechanical testing. And then we split it into 4 groups. So one was elastase, where you can use this protease called elastase, and we did a pretty brief treatment just for 6 minutes, and it will degrade the elastic fibers in the wall. And then we did PGG alone, because we wanted to make sure that PGG didn't change the mechanics. And then we did what we call a preventative, this one is the restorative treatment. So ELA first, so degrade and then treat with PGG, or preventative, where it's PGG first and then ELA. Then we did the mechanics all over again. And then we used constitutive modeling, so to understand the complete biaxial mechanics in terms of any stretch and stress protocols. So if we just focus on a single protocol first, right, so back to this, we stretch it to its in vivo length, and then we inflate it. So untreated and PGG are here in gray and yellow, and you can see that PGG alone doesn't do much to the pressure diameter behavior of the aorta. Elastase is here, right, so huge increase in the diameter at low pressure, so basically it can't resist deformation at all at low pressures because we've affected the elastic fibers. Collagen is still intact, and you can see that it stiffens way earlier than it would in an untreated aorta. And then our treatments are somewhere in between. So preventative, which is navy blue, is closer to untreated than restorative, which is kind of this turquoise color. But both of them bring the mechanics back towards the untreated condition. In the axial direction, this is the slope. This is the compliance. So if you just take the slope of this curve, it's the compliance. And in untreated aorta, we always see this behavior that the compliance peaks somewhere near physiologic pressures. So your aorta is designed to have peak compliance here. And when you treat it with elastase, it's at really low compliance here. And again, our treatments are somewhere in between, with preventative being better than restorative. If we then look at the stretch and the stress behavior, right, which is now not dependent on the geometry or the thickness changes that could be happening, here's our behavior in the untreated and the PGG. They're right on top of each other. The elastin has no resistance, or elastase has no resistance here, and then very stiff. And our treatments are somewhere in between, with the preventative being closest to the untreated. We are interested in the biaxial mechanics in this case, because we know that elastin can actually contribute quite significantly to axial behavior of the vessel. So in the axial direction, the treatment is not as effective. Here's our untreated right here. Here's the elastase down here, and then our treatments are not the same as the elastase. them to do exactly the loading conditions we might see in vivo. And so that's where we use constitutive modeling. So we use a model called Wagenseil, which assumes that the aorta is like a fiber reinforced composite. So you have the fibers, which are shown here, which are modeled as the collagen fibers, and then they're sitting in this like isotropic matrix that we use in neo-Hookean type of material form, which is similar to a rubber-like material. And so by using this model, we can then fit parameters and try to understand how things might change between like what you might consider the neo-Hookean matrix associated with elastin and with these fibers that are associated with collagen. So when we do this, these are the representative parameters that we get for our 5 groups. So first thing, the R-squared is a measure of how good this model fits our data. Untreated actually fits pretty good. Elastase fits terribly. Right? So this is not a good R-squared. It does not fit well. But we actually thought that was interesting because it shows how much the elastase changes the mechanical properties that you really can't use this model we would normally use to fit the data very well. And then when we use our different drugs, it comes back towards fitting like similar to an untreated. And then you can look at the parameters, and the C1, which is associated with that neo-Hookean matrix, really drops a lot when we degrade the elastic fibers. Even in our treatments, it's still degraded quite a bit. But then it kind of, the behavior comes back with various changes to these nonlinear fibers. But now that we have these parameters, we could go predict how the aorta might behave under any loading. We don't have to just go with the experimental loading that we did. And in this case, we're looking at strain energy, so not stress, but strain energy, which is related to how much that wall can store. We need to form it, how much energy it stores that can then be used to do work on the blood. If you think about pulling a rubber band and you let go and it snaps back, it hurts your fingers because it stored energy, which then is used and you can feel that, okay? And so in terms of the strain energy, I'm now blocking off the physiologic region. And what you can see is that our untreated, our PGG, and then our preventative treatment all have approximately the same strain energy under an equal biaxial loading protocol where both circumferential and axial directions are the same. Our elastase is way down here, and then the restorative is down here as well. And so by using this drug, we have restored kind of that strain energy storage in the wall, which is really important for cardiovascular function. We did our multiphoton imaging to see what we were actually doing to the wall, and we were quite surprised that in the elastase condition, right, that has huge changes in mechanics, it doesn't actually look that bad, okay? So there are a few extra little fragmentations in the elastic fibers, some buckling that you see between the elastic fibers and the collagen fibers that might be related to changes in how they're connected. But the point of this is there's pretty subtle changes in structure that are leading to big changes in mechanics. This buckling you don't see it in our preventative treatment, you see it a little bit in our restorative treatment. And then we also noticed in the PGG these little like adhesions where the arrow is, where it looks like those elastic laminae are stuck together. We don't know exactly what that means, but we do want to be careful with this drug treatment because we don't want anything that's going to change the structure in just the normal case. But in the future, so this was just in an in vitro model, right? We put the PGG and the elastase through the lumen, which is how you might treat a human by delivering it intravenously. But none of this was in vivo. It is known that PGG has additional effects in vivo. It is supposed to help with the formation of new elastic fibers. It is supposed to be anti-inflammatory. And so this might all improve its efficacy when we look at it with live cells. We're also interested in genetic TAA models. So we have these models like the Marca mouse that I showed you that has TAA. And we'd like to know if this PGG might work in these animals. We're also interested in how these changes in mechanical behavior that we can measure, right? So both with and without the elastic fiber fragmentation affect other wall stresses. So far I've shown you pretty simple measures of wall stresses, but we can measure more complicated ones as well as shear stresses. So a little bit of preliminary data on some of these bullet points. And one is looking at in vivo delivery of PGG. So this work is in collaboration with Nara Bahare at Clemson, and he has put PGG into nanoparticles. The nanoparticles have an antibody to elastin that targets elastin after you get to in an abdominal aortic aneurysm model, that if you put a fluorescent tag in these nanoparticles, then it will go to the site of the aneurysm in a rat. He also showed in the rat model that the PGG was effective in what I would call a restorative fashion. And so in this case, they put like elastase or something on the rat to start an aneurysm, and then they started treating it with the PGG nanoparticles, and the aneurysm stopped dilating, whereas in all the other cases, it continues to dilate. But still, these are chemical models of aneurysms, so we were really interested in these genetic models. And so far, what we've done is we have given our Marfan mice, so the mutant mice that I showed you before, we've given them nanoparticles that have the fluorescent tag inside. And in this case, so this is the mutant mouse that has an aortic aneurysm and it has the nanoparticles. You can see that they go towards the ascending aorta, which is dilated. In a heterozygous mouse with the nanoparticles, no signal. Wild type mouse with the nanoparticles, no signal. And then this is a wild type mouse that was injected with saline. However, we know that all of the aorta has some sort of elastic fiber fragmentation in our mutant mice. So why is it only going to that aneurysm? So it turns out that you crank up the fluorescence, right? Anyone that's done fluorescence microscopy, you crank up the gain, you kind of see whatever you want, right? So you crank up the gain in this case, and you can see that this fluorescence shows up throughout the aorta in the mutants. It also shows up throughout the aorta in a heterozygous and a wild type. These nanoparticles with the elastin antibody are sticking to the wall, right? Much less in where we don't expect fragmentation, or not as much fragmentation compared to this aneurysm region where I showed you big fragmentation. And then if you just give them saline, right, there's nothing there to stick, but we've cranked it up enough that we do have some artifacts. And so we do think these nanoparticles can get to where we want them to go. We think that they're going to go more towards the aneurysm than to other parts. So we're treating these mice right now to see if the PGG might be able to prevent the dilation and the outcomes. I also mentioned the wall stresses and mentioned that a lot of what I've shown you is pretty simplified calculations of wall stresses. So in collaboration with Victor Barocas at Minnesota, we do fluid structure interaction modeling to understand the solid and fluid-induced wall stresses. So for this work, we take an MRI of our mice. We do MRI at various time points so that we can look at the aneurysm formation over time. We then isolate the aorta. We've done some things like we think tortuosity might play a role. So this is looking at the geometric length over the actual length and looking at tortuosity in the mice. We then do all our mechanical testing so we can get mechanical properties. And then we can look at other measures of stress beyond just that simplified axial and circumferential, such as the von Mises stress that is plotted here. If we know fluid boundary conditions, then we can also look at some of the shear stresses. So in this case, here's our aorta. We need to know what's happening at the inlet. And so we need to prescribe a flow rate coming in from the heart. We need to know what's happening at all the outlets. And so we use these resistance and capacitance models to dictate how the pressure and flow change at different places. And then this is our Holtzapfel, Gassler, Ogden constitutive model, because we need to know how the wall properties, what the wall properties are. So using these types of models, we can plot all kinds of biomechanical parameters. In this case, we're looking at oscillatory shear stress. So these are 10 different mice, 10 different supposedly genetically identical mice, some males, some females. And we were looking at lifespan. So this is a fibulin 4 mutation that gets throughout the aortic aneurysm. And they live a lot longer, so you can see the earliest time point is 12 months. The latest time points are 25 months, and that's because we needed to end the study. So we were done. So those mice all got sacked at 25 months. That's the longest lifespan we allowed them to have. So statistically, that then gave us some heartache doing all our statistics, but there are ways that you can account for that in your statistics. So we had to have a statistical consultant to look at this data. But what you can see is a wide variety of aneurysm phenotypes, right? And some, like this one, seems to have a pretty good aneurysm, dies at 12 months. This one, but then the next one, you're not as big. And then some of them that live a long time have like almost no aneurysm, and some, have a big aneurysm. So we were interested in like how do all of these biomechanical or biofluidic biomarkers correlate with outcomes, and how do they compare with what surgeons actually use, which is just max diameter. So this is a whole correlation matrix where we have things like max pressure, we have von Mises stresses, we have that arterial tortuosity index, a bunch of measures of oscillatory shear index, Reynolds number, time average wall shear stress, Wagenseil number, and then diameter max and lifespan. So hopefully you can see the little black boxes down here. So here's D max with lifespan. So there's actually two biofluidic biomarkers that correlate better with lifespan than D max. And those are what are plotted here. So it's oscillatory shear index, and then the time average wall shear stress minimum. So both of those correlate better with lifespan than D max, which was currently used. And so we're at the point now with imaging and with some of the computational power that you could envision someday you could make a patient-specific model and calculate these parameters and get an idea of their risk, right? And so we're now using this with human imaging data to see if we can get some of these same biomarkers. And so in this case, we're tracking some of these biofluidic biomarkers over time. We're calculating them in additional mouse TAA models, like this Marfan model that has a much more aggressive form of the thoracic aortic aneurysm. We want to understand how those different wall stresses might contribute to the growth, remodeling, and failure. And then, as I mentioned, we're working with human TAA. So at WashU, there's a clinic for genetic aneurysms. And so they have over 700 patients that have been imaged multiple times. So we're working with some of that imaging data to try to do some of the same modeling and see if some of these biofluidic biomarkers might be predictive of their outcomes. Okay, so I started with wall stresses, but now I want to talk a little bit about transmural transport. So we measure fluid and solute transport using the same system that I showed you before, but we just close off one end and we use a static pressure column. So if this is a closed system, we put a bubble here. If the bubble moves, that means fluid has gone somewhere. So we assume the fluid's going across the aortic wall. And then we do the experiment again with a fluorescent tracer in here. And if the fluorescence in the bath increases, that means that tracer has gone across the wall. So we can measure fluid flux and we can measure solute flux. We can then use some 1D infection diffusion equations to calculate two things: hydraulic conductance and solute permeability. Hydraulic conductance is related to the fluid flux, solute permeability is related to the solute flux. We then use some of these experiments to try to understand what might be happening in another aneurysm model, the fibulin 4 knock-in mice. These have a missense mutation, so an E is turned to a K at position 57, was identified in humans and then modeled in mice. And similar to what I kind of showed you with like varied phenotypes, these mice, so these two groups are genetically identical, and one has no aneurysm or NA, and one has an aneurysm. So about 50% of the mice get an aneurysm, and we don't know what. So we really wanted a more consistent aneurysm phenotype, and so we bred them to lysyl oxidase heterozygous mice, which I showed you in the beginning was one of those cross-linking proteins. And so if you remove some of the cross-linking enzyme, then they all get aneurysms. So with these different genotypes, we have this kind of continuum of aneurysm dilation. So you can see the diameter goes up depending on kind of which group you're in. So we then did all our transport measurements. And we were looking at hydraulic conductance first, right, which is the ease of fluid movement across the wall. We do it without our tracer, and then we do it with our tracer and get similar results, where it's going down as aneurysm phenotype gets worse. So hydraulic conductance decreases. In terms of solute permeability, right, which is the ease of the solute moving across the wall, it goes up in our non-aneurysm, so they have a genetic mutation, but they don't have aneurysms. And then it goes down when you have an aneurysm, so up and down. Both of these are totally opposite to our hypothesis. So we thought if you fragmented elastic fibers, things would go across easier, right? So the hydraulic conductance would go up, so easier to go across the wall, and solute permeability would go up, easier for the solute to get across the wall. So not what happened, but it was a good lesson. So what the heck is happening? Why are these things going in the direction we're going? We had to look at the wall, so we're okay, like, do we really have elastic fiber fragmentation? What's happening? These are these multi-photon images again, and you can see kind of these breaks and less intense staining in our aneurysm models. If you quantify endpoints, you can see they all have breaks. It doesn't seem to correlate with aneurysm phenotype in this case, so they just all have breaks. So then we were trying to think about what else could be happening, and it turns out there's deposition of other matrix proteins besides elastin and collagen in aneurysm models, and in particular, proteoglycans have been shown to be increased. So in this image, this is a histology image where we're staining for a whole, or not staining, but the chemicals used highlight a whole bunch of different things, and proteoglycans are in turquoise. So they're not usually in the wall. They're these highly charged, highly hydrated molecules that are mostly in things like cartilage, not usually in your aorta, but they show up in aneurysms. And we think that what could be happening is if that gets deposited, they're highly hydrated, highly charged, they could be influencing the way that both fluid and solute move across the wall and changing these parameters that we were measuring. The other thing is that those parameters we were measuring are something like a structural transport property. So they depend on geometry, they depend on mechanics of the wall, they depend on thickness. And so you actually need more sophisticated models if you want to look at transport material properties that are independent of those things. And so we can do multiphasic modeling with our data to get to material properties of transport, which is hydraulic permeability and effective diffusivity. And so in this case, we have a whole bunch of experimental stuff that needs to go in. We have, we're using FIU to do multiphasic and biphasic continuum modeling, and then we're getting out these model outputs. Of course, we need to know all of these things over here. So we need to know like the material properties. And so we use both our data and some published data to understand what the diameter would be at different pressures, and then also what the stress strain curves look like. And then we did some work that I'm going to skip over in the interest of time, looking at parameter sweeps on what's important. So we'll skip our parameter sweeps and we'll go to the punchline, which is these material transport properties. So this hydraulic permeability is the material version of the hydraulic conductivity. And you can still see it's still decreasing with our different phenotypes, but not quite as much as it was. So accounting for all of these other things didn't change the results, but it does tell you that we're definitely changing material properties as this aneurysm gets worse, and that our model does a pretty good job of predicting experiments. And then that effective diffusivity is the thing that is going up and then down, and we still get that behavior once we control for all of these other variables like changes in mechanics and geometry. And our model fits pretty well. And so we think that what could be happening in this transport is, we have these initial defects, we know that there's elastic fiber fragmentation, but if that actually increases the hydraulic permeability and effective diffusivity, allowing the aberrant cytokine deposition, so in the early stages it is increased like we expected, but then what happens is you get remodeling of the wall, proteoglycan deposition that then actually decreases it. And so we are interested to know if we could track these transport changes throughout TAA progression, rather than in these separate genetic models, but in like the MGR model, the Markov model over time. How does that correlate with the progression in the model? We think that there might be things in your blood that have altered transport in TAA, and could we use those either as like biomarkers of the disease, or if those things are getting into the wall and contributing to the pathogenesis, could we think of a way to stop that? We're interested in measuring transport in human TAA. So if people get surgery and their ascending aorta is replaced, we can get that tissue. So we're interested in measuring the transport in that tissue, and then using that to stage, diagnose, or treat TAA. So we've done a little bit of work in our Markov model that I'm going to skip. I'm just going to go to what we're doing in the lab in general. We're looking at these sex differences in the TAA outcomes. We're looking at trying to treat the mice in vivo to stop this elastic fiber fragmentation. Looking at these fluid and structure-induced wall stresses in both mice and humans. Looking at the transport. And then we have a whole other arm of the lab that's actually looking at supravalvular aortic stenosis, which is a genetic mutation that reduces elastin amounts completely. It actually gets an opposite phenotype, so narrowing in stenosis instead of aneurysm. So we're really interested in like why defects in elastic fibers sometimes lead to very opposite remodeling of the aortic wall. And I mentioned that it's, not sure why that mentioned that the work is all done in the lab, so when I say we, right, that's the royal we. I don't actually do these things, I just talk about them. But the people that do this work in particular today have been circled, so Keshav and Prashan, and then Christine, the former lab member, did a lot of the work that's shown in here, and my collaborations and funding, which hopefully will keep going. So thank you for your attention.
Thank you so much for this amazing presentation. So we work on Alport syndrome, which is a collagen for every mutation caused disease, and it's a rare CKD disease. And the mouse model is amazing. It mimics the renal function, the ocular, the auditory, all of those phenotypes are there, except for aortic aneurysm or aortic rupture.
Okay.
So as I was listening to this presentation, I was thinking, do you think if we cross the mouse with the locks, heterozygous knockout, we could get that?
So there's many examples in the mice where you have to do something a little more aggressive genetically to get the aneurysm phenotype. So we have seen, in this case, we crossed it with the lysoxidase het. We've seen ones where sometimes the phenotype is only in a homozygous mouse, whereas the human phenotype is heterozygous. We've seen things where people do like perhaps tube delivery or something to increase blood pressure, and then they get an aneurysm. And so, mice and humans are different, and there's many examples where in a mouse, the exact same genetic insult in a human does not give you the aneurysm of a mouse, but pushing it a little bit will. So yeah, we have to talk.
So do you think that's the right place to start? Or?
I think that's certainly not a bad strategy.
Okay, thank you.
Yeah.
Last one quickly. I was surprised to see in the early slides you showed with gene mutation that lower stiffness in the low loading region,
Yeah.
right? So the elastomers are gone.
Thanks. That suggests that the collagen fibers were being crimped, I
Yeah.
guess, in those cases, and what's keeping them crimped?
Yeah. So I don't know exactly, but it's pretty parallel to what we see with elastase, right? So with elastase, you lose that kind of resistance to deformation in the low region. They kick in earlier, right? So if you look at an elastase curve, it gets stiff at more like 50 millimeters of mercury instead of 100. And so I think a lot of it depends on exactly where you make that cutoff. So I think in the very low, which is where we're fitting, the collagen has not kicked in yet, but I think it does probably kick in earlier.
And then the sex bit differences, do we observe something similar in humans?
So in humans, there's no difference in men or women having Marfan, but outcomes are different. Yes. So males are more likely to have a cardiovascular event leading to adverse outcomes. And no one really knows why, but it might have something to do with remodeling the wall.
Any last questions from the audience? I hear a bunch of buzz outside, so thinks the high schoolers might be here.
It's time.
So we're going to move on from here. Dr. Wagenseil will be around all day. If you have questions for her that you don't want to ask from everyone, feel free to come to her throughout the day. If you haven't set up your posters yet, please do so. Those of you who are involved in the outreach event, that's going to be upstairs on the 3rd floor. Otherwise, mingle around the posters. If you are judging, please come see me ASAP. Thank you.